Hi, I wanted to recap some of the stuff, main points from class from this week, and then also discuss a little bit about upcoming topics as well. So first of all, we just talked about the general parts of a neuron, the cell body, and dendrites. Dendrites are, of course, where inputs from other axons come in. We'll be talking a lot more about these synaptic contacts really all semester, but especially starting next week. We also discussed the axon and the idea of an action potential. So this is a signal that's generated in the cell body that travels along the axon, and then it gets to the ends of the axon. It's called presynaptic terminal. It's where this sending neuron, the neuron that's created the signal that's going to send a signal onto the next neuron, called the presynaptic neuron, releases a chemical called a neurotransmitter, and then that chemical diffuses across the synaptic connection and will bind to receptors on the receiving cell or the postsynaptic cell. So before the synapse is the space between them. Before the synapse is the sending neuron. After the synapse is the receiving neuron, so pre and postsynaptic. At the receiving end, there will be proteins that sense the chemical neurotransmitter, bind to it, and open ion channels that allow sodium ions or other ions to flow through. We discussed quite a bit on the first day about the electrical and salt situation on the neuron. So there's a protein called the sodium and potassium pump that uses ATP to force sodium out and pull potassium in. This creates an unequal balance of ions across the cell, where there is a higher concentration of sodium out versus in, and a higher concentration of potassium in versus out. There are also leak potassium channels. These are channels that are always open, and so a little bit of potassium is always leaking out of the cell. What that means is that inside the cell, if nothing else is going on, the cell's just hanging out, then the voltage inside is going to be negative with respect to the outside. If then the cell gets sufficient amount of excitation, meaning that neurotransmitters are released onto its dendrites, sodium flows into its dendrites, the inside of the cell gets more positive, that can cause a little blip, or if it gets up to a threshold point, then other channels called voltage-activated sodium channels open, and a very quick inflow of sodium comes in. This causes a very rapid depolarization, positive charge inside the cell, and then at the peak of this, the sodium channels will close, stop passing current, and potassium channels open. And then a bunch of potassium ions quickly flow out, and then the cell actually ends up a little bit more negative than it was at rest. It's called the after hyperpolarization. And then over the next few milliseconds, the cell will reset itself. A slightly better view of this that is a little bit more to scale is here, where it shows very clearly that the depolarization from the synapses is slower than this rapid rise during the action potential itself. So then today, let's see, and then we also discussed just in terms of the sequence of events in the action potential. And so this and sort of the inputs going on and so on. The action potential itself is this sort of steps 3 through 5, from the moment it reaches threshold until the moment the cell returns back to, until the cell gets to its after hyperpolarization. And then step 6 is sort of is called the refractory period. So it's a period after the action potential is over, when the cell is not yet quite ready to fire a second action potential. Steps 1 and 2 here are what's leading up to the action potential, and we're going to go this next week more into this sort of between step 1 and step 2. Today, on Thursday, we discussed how these action potentials travel along the axon. And so there's quite a lot going on here. The electrical signal, because the electrical signal dies off over space, what that means is it needs to be remade again and again and again and again and again along the length of an axon. And in a bare axon without any insulation over it, then the electrical signal just makes it a short distance before it's gotten too weak to get the next segment of the axon to reach its own threshold. So we talked about how one segment, how the cell body reaches threshold, and then a half millisecond later the cell body's at the peak, and then the first segment of the axon reaches threshold. Then a half millisecond later, the cell body's in a refractory period. The first segment of our action potential is at its peak, and the second segment of our action potential has reached its threshold. A half millisecond later, the cell body's starting to come out of its refractory period. The first segment of our axon is in the refractory period. Our second segment has now reached its peak, and our third segment is now at threshold. And this has to get remade every half millimeter or so, and it takes about a half millisecond per half millimeter, which is kind of fast, like as fast as a car drives, but it is, for communicating signals along your body, it's way too slow. And so in order to solve this problem of these signals being too slow, neurons insulate the axon. What the insulation does is it means that the electrical signal travels farther before it reaches a point that it's not strong enough. And so now here our cell body, or at the beginning of our axon, reaches threshold. And then at that point in time, the first node of Ranvier is not yet at threshold. Then our cell body fires an action potential, and our first node of Ranvier, here, our first space between these insulated myelins, has reached threshold. Come back a half millisecond later, and now this first segment of the axon will be in a refractory period. This first node here will be at its peak, and our second node will be reaching threshold. And so what that means is that our signal, instead of being made every half millimeter, it's remade every 50 millimeters. And as a result of that, it's getting remade 100 times less often, and therefore traveling 100 times faster. So now the signals travel 100 meters per second. We then talked about the cells that make the myelin in the central nervous system. They're oligodendrocytes. They reach out and wrap their membrane around, they wrap the whole cell in the membrane around the axon a bunch of times. And since membrane is fat, that's a lot of thick insulation. If the myelin goes away, then this is what happens in multiple sclerosis and also something else called Guillain-Barré we'll talk about on Tuesday. But if the signal goes away, then the signal might not reach the end. If that's in a sensory system, that might be perceived as numbness. Also, as these axons are starting to lose their myelin, there are sometimes random action potentials that don't correlate with something. And so that can cause sort of like sensory illusions, tingling sensations for touch. If this is multiple sclerosis, and it affects, so we also talked today about, what is the central nervous system? The central nervous system is the brain and spinal cord. In multiple sclerosis, then the myelin in the central nervous system is what's breaking down. This is seen as, so this is stuff that we're going to talk about on Tuesday, but it's seen as like spots in the axon areas in the brain where there are hardenings, where there's a bunch of dead axons. And as a result of that, you can have sensory problems and motor problems if if the axons in your spinal cord that are bringing in touch information or bringing out motor information are breaking down. But you can also have other problems if the communication failures are happening in the brain. It can lead to feelings of fatigue, feelings of depression, changes in mood, changes in cognition, as well as weakness. And then again, because of these unintended random action potentials, muscle spasms, or loss of sensation, hypothesias, or unusual sensations, pain or paresthesias, as well as other problems in terms of speech, visual, auditory, and so on. So there are many different things that multiple sclerosis can cause. Next class period, we're also going to talk about Schwann cells and myelination in the peripheral nervous system, but also leading up to next week, the other thing that we're going to be talking about is synaptic communication and presynaptic terminals, presynaptic axons, and the release of neurotransmitter. And for that, there's another video up on Canvas that you should watch in preparation for the homework, which is going to ask about that for next week. Also, one other thing that we discussed in class today was in general about reading papers, and then in particular about the this one paper by Tsutsui talking about investigating myelination in the peripheral nervous system. And so, um, uh, so as with most of our papers, really start with the abstract and what we talked about with this particular study, or actually, I guess, sort of more generally, with all papers, you should be thinking about what is the question, what are the possible results, what are the manipulations, what are the measurements, and making very clear the difference between what they do, the manipulation, and what they observe, the measurements. The expected results in the form of if, then, and then because of some invisible process, and then the results, which is going to be really one of our possible observations, and then our conclusion, which really follows from this sort of because invisible process, what we think is going on. For some of the papers, we're also going to be talking about follow-ups. In this particular study, as we work through the abstract, there are a few questions that they're asking, but one of them is, does this particular TRPM2 channel affect immune cell migration into the central nervous system, and how does that influence multiple sclerosis symptoms? And so generally, there's sort of a couple possibilities. For some papers, we'll go a little bit more. There might be 3 or 4 possibilities, but either this channel is or is not stimulating the immune cells moving into the central nervous system. The manipulation, they had 2 different things they do. They have a whole bunch of mice, and all of the mice, they give them a series of injections that creates an immune response that mimics multiple sclerosis symptoms. Then and then in half of those mice, they were genetically normal. The other half of those mice had been previously genetically engineered to be missing the gene for this one particular channel that they're interested in. So all of the mice have the series of injection. Half of them are genetically normal mice. The other half are mice that are missing this channel. Those are the manipulations. That's what they did to the animals. What they're then measuring is they're measuring how many immune cells are finding their way into the central nervous system. In terms of their possible results, so if this channel is altering immune infiltration, then they would expect that when you get rid of the channel, there are going to be fewer immune cells. That's the observation. That's actually what they're going to see in this knockout mice. And then the invisible process that they aren't measuring is that this channel is stimulating these cells to enter into the central nervous system. The alternative possibility, we went a little bit quickly over this, but if there's no influence of this channel, then they would expect the normal mice and the mice that are missing this channel, so-called knockout mice, to have the same number, because this channel isn't involved in this infiltration. So the result then was fewer immune cells observed, and actually one other thing to point out, sorry, is that all of this came from just the abstract. We were able to get this far just with the abstract. And then in terms of the actual results, as with many studies, there are many different measurements and many different experiments. They did some experiments about how this affects behavioral aspects of multiple sclerosis. They did experiments about how this affects T cells, and the T cells turned out not to be affected. And then the one that they were interested in is so-called monocytes, which is another kind of immune cell. There are a few different measures of monocytes. I just showed one, these IBA1 cells. If we were really writing a report about this, we would have a couple sentences explaining what IBA1 is and so on. But the main observation here is that in mice that are normal, that are wild type, that don't have any genetic changes, so they've got the normal sort of genetic makeup, after the series of injections, a whole bunch of immune cells get into their brains. But in these knockout mice, the series of injections still causes immune cells to come into the brain, but it is significantly fewer. So that is their observation, and then their conclusion essentially follows again from this because statement that we had here. There's some invisible process where the TRPM2 channel, when it's there, it helps the immune systems get in, and that's why they observed that the animals that are missing this channel don't have as many immune cells getting in. Like I said, you should also watch the video about synaptic transmission on Canvas as you're getting ready for your homework, and then we'll review, keep talking a little bit more about this study on Tuesday. talk about Guillain-Barré syndrome, and then Tuesday and Thursday of next week we'll be talking about synaptic communication.